Good day, everyone. Our topic for today is all about fungi. Our learning objectives for today is to list the defining characteristic of fungi, differentiate asexual from sexual reproduction, and describe each of the processes in fungi, list the defining characteristic of the four phyla of fungi, and identify the two beneficial and two harmful effects of fungi. Fungi are in the fungi kingdom. They are chemoheterotrophs and acquire food by absorption. With the exception of yeast, fungi are multicellular. Most reproduce with sexual and asexual spores. Of the more than 100 species of fungi, only about 200 are pathogenic to humans and animals. However, over the last 10 years, the incidence of serious fungal infection has been increasing. These infections are occurring in healthcare settings and in people with compromised immune systems. These infections are often caused by fungi normally found in and on the human body. In addition, thousands of fungal diseases affect economically important plants, costing more than $1 billion annually. Fungi are also beneficial. They are important in the food chain because they decompose dead plant matter, thereby recycling vital elements, the hard parts of the plants, which animals can't digest, are decomposed primarily by the fungi through the use of extracellular enzymes, such as cellulases. Nearly all plants depend on the symbiotic fungi, known as mycorrhizae, which help the roots absorb mineral and water from the soil. Fungi are also valuable to animals. Fungi farming ants cultivate fungi that breaks down cellulose and lignin from plants, providing glucose that the ants can then digest. Humans use fungi for food like mushrooms, and to produce foods like bread and citric acid, and for drugs, alcohol, and penicillin. The study of fungi is called mycology. A pathogen must be accurately identified if the disease is to be properly treated and its spread prevented. We will first look at the structures that are the basis of fungal identification in clinical laboratory. Then we will explore their life cycle and nutritional needs. All fungi are chemoheterotrophs requiring organic compounds for energy and carbon. Fungi are aerobic or facultatively anaerobic. Only a few anaerobic fungi are known. Characteristics of fungi. Identifying yeast in bacteria requires biochemical tests. However, multicellular fungi identified on the basis of the physical appearance, including the colony characteristics and the reproductive spores. Vegetative structures, fungal colonies, are described as vegetative structures because they are composed of cells involved in catabolism and growth. Molds and fleshy fungi, the thallus or the body of a mold or fleshy fungus, consists of long filaments of cells joined together. These filaments are called hyphae, and it can grow into immense proportions. In most molds, the hyphae contains cross-wall called septa, which divide them into distinct unicellular cell-like units. These hyphae are called septate hyphae. In few classes of fungi, the hyphae contains no septa and appear as long, continuous cells with many nuclei. These are called syncytic hyphae. Even in fungi with septate hyphae, there are usually openings in the septa that makes the cytoplasm adjacent cells continuous. These fungi are actually syncytic organisms too. Hyphae grow by elongating at the tips. Each part of the hypha is capable of growing, and when the fragment breaks off, it can elongate to form a new hypha. In the laboratory, fungi are usually grown from the fragments obtained from a fungal thallus. The portion of the hypha is obtains nutrient is called the vegetative hypha. The portion concerned with the reproduction is the reproductive or aerial hypha, so named because it projects above the surface of the medium on which the fungus is growing. Aerial hyphae often bear reproductive spores. Discussed later, when the environmental conditions are suitable, the hyphae grows to form a filamous mass called mycelium, which is visible to the unaided eye. These pictures shows the different structures of a fungi, namely the septate hypha, sinusitic hypa, and the growth of the hypa from the spores. And these pictures from below shows the aerial hypae and vegetative hypae. Yeast. Yeasts are non-filamentous unicellular fungi that are typically spherical or oval, like molds. Yeasts are widely distributed in nature. They are frequently found in white powdery coating on fruits and leaves. Budding yeast such as Saccharomyces divide unevenly. In budding, the parent cell forms protuberance or buds on its outer surface. As the bud elongates, the parent cell's nucleus divides, and one nucleus migrates into the bud. The cell wall material is laid down between the bud and parent cell, and the bud eventually breaks away. One yeast cell can in time produce up to 24 daughter cells by budding. Some yeasts produce buds that fail to detach themselves. These buds form a short chain of cells called pseudohypha. Yeasts are capable of facultative anaerobic growth, which allows the fungi to survive in various environments. If given access to oxygen, yeasts perform aerobic respiration to metabolize carbohydrates into carbon dioxide and water. Denied oxygen, they ferment carbohydrates and produce ethanol and carbon dioxide. This fermentation is used in the brewing of wine, making and baking industries. Saccharomyces spices produce ethanol in brewed beverages and carbon dioxide for leavening bread dough. Dimorphic fungi. Some fungi, most notably the pathogenic species, exhibit dimorphism. Two forms of growth, such as fungi, can grow either as a mold or as a yeast. The mold-like forms produce vegetative and aerial hypae. The yeast-like forms reproduce by budding. Dimorphism and pathogenic fungi is temperature dependent. At 37 degrees Celsius in the fungus is yeast-like, and at 25 degrees Celsius it is mold-like. However, the appearance of the dimorphic in the instance of non-pathogenic fungus shown in figure 12.5 changes with CO2 concentration. Life cycle. Filamentous fungi can reproduce asexually by fragmentation of their hypae. In addition, both sexual and asexual reproduction in fungi occurs by the formation of spores. In fact, fungi are usually identified by spore type. Fungal spores, however, are quite different from bacterial endospores. Bacterial endospores allow single vegetative bacterial cell form one endospore, which eventually germinates to produce a single vegetative bacterial cell. This process isn't reproduction because it doesn't increase the total number of bacterial cells, but after a mold forms a spore detaches from the parent and germinates into a new mold. Unlike the bacterial endospore, this true reproductive spore, a second organism, grows from a spore. Although fungal spores can survive for extended periods in dry or hot environments, most do not exhibit the extreme tolerance and longevity of bacterial endospores. Spores are formed from aerial hypae in a number of different ways, depending on the species. Fungal spores can be either asexual or sexual. Asexual spores are formed by the hypae of the one organism. When these spores germinate, they become organisms that are genetically identical to the parent. Sexual spores results from the fusion of nuclei from two opposite mating strains of the same species of fungus. Asexual spores are produced by an individual fungus through mitosis and subsequent cell division. There is no fusion of the nuclei of cells. Two types of asexual spores are produced by one fungi. One type is conidiospore and the other one is conidium, or a unicellular or multicellular spore that is not enclosed in a sac. Down below are sample images of asexual spores. Sexual spores are fungal sexual spore results from asexual reproduction, which consists of three phases. First is the plasmogamy, which a haploid nucleus of a donor cell positive penetrates the cytoplasm of a recipient cell negative. Next is the chirogamy, which is the positive and the negative nuclei fuse to form a diploid zygote nucleus. Third is the meiosis. The diploid nucleus gives rise to the haploid nuclei sexual spores, some of which may be genetic recombinants, and the sexual spores produce the fungi characterized by the phyla. Clinical identification is based on the microscopic examination of sexual spores because of most fungi exhibit only a sexual spore in laboratory setting. And on this image, you can see the asexual reproduction and the sexual reproduction of a spore. Nutritional adaptations. Fungi usually grow better in an environment with a pH of about 5, which is too acidic for the growth of most common bacteria. Almost all molds are aerobic. Most yeasts are facultative anaerobes. Most fungi are more resistant to osmotic pressure than bacteria. Most can therefore grow in relatively high sugar or salt concentrations. Fungi can grow on substances with very low moisture content, generally too low to support the growth of bacteria. Fungi require somewhat less nitrogen than bacteria for an equivalent amount of growth, and fungi are often capable of metabolizing complex carbohydrates such as lignin that most bacteria can't use for nutrients. These characteristics enable fungi to grow on such unlikely substrates as bathroom walls, shoe leather, and discarded newspapers. Medically important fungi. This section provides an overview of medically important phyla of fungi. The actual diseases they cause will be studied in Chapter 21 and 26. Note that the only relatively small amount of fungi cause diseases and some fungi protects us from the infection. Here are some lists of fungi that can cause human diseases. First is Zygomycota, next is Microsporidia, next is Ascomycota, and the last but not the least is Basidiomycota. The fungi we have looked at thus far are telomorphs, that is, they produce both sexual and asexual spores. Some ascomycetes have lost the ability to reproduce sexually. These asexual fungi are called anomorphs. Both telomorphic and anomorphic names are given for some of the fungi because some medically important fungi are known by their anomorphs or asexual names. Fungal diseases. Any fungal infection is called mycosis. Mycoses are generally chronic or long-lasting infection because fungi grow slowly. Mycoses are classified into 5 groups according to the degree of tissue involvement and mode of entry into the host. First is the systematic, subcutaneous, cutaneous, superficial, and opportunistic. Fungi are related to animals. Consequently, drugs that affect fungal cells may also affect animal cells, making fungal infections of humans and other animals often difficult to treat. Some fungi cause diseases by producing toxins. Attached here is a picture of the characteristics of some pathogenic fungi. The economic effects of fungi. Fungi has been used in biotechnology for many years. Aspergillus niger, for example, has been used to produce the citric acid for foods and beverages since 1914. The yeast Saccharomyces cerevisiae is used to make bread and wine. It is also genetically modified to produce a variety of proteins, including the hepatitis B vaccine. Microbiome. Just as viruses and bacteria are members of a healthy human microbiome, so are fungi. But unlikely the bacterial portion of the microbiome, the so-called microbiome is just beginning to be studied. Members of the yeast genus Candida are the most common fungi that lives as normal microbiodata in the mouth, intestine, and vagina. The most diverse population of fungi is found in the mouth where 101 species have been identified. Understanding how fungi among the microbiome interact with each other is important. The next topic is all about lichens. A lichen is a combination of green alga and fungus. Lichens are placed in kingdom fungi and are classified according to the fungal partner most often as an ascomycete. The two organisms exist in a mutualistic relationship in which each partner benefits. Lichen is very different from either alga or fungus growing alone, and if the partners are separated, the lichen no longer exists. Approximately 13,500 species of lichen occupy quite diverse habitats because they can inhabit areas in which neither fungi nor algae could survive alone. The lichen's medulla is composed of fungal hyphae surrounding the algal layer. The protective cortex is a layer of fungal hyphae that covers the surface and sometimes the bottom of the lichen. Lichens can be grouped into 3 morphologic categories: the crustose, foliose, and fruticose. Importance of lichens. Lichens has considerable economic importance in ancient Greece and other parts of Europe as dyes for clothing. Arsenic acid from Urcea is used as an antimicrobial agent in China. Erythrolitmin, the dye used in litmus paper to indicate changes in pH, is extracted from a variety of lichens. Lastly, lichens are major food for tundra herbivores such as carabao and reindeer.